Hello friends, welcome to my YouTube channel, the Microbiology Digest. Today again, I am back with a new topic. It is bacterial genetics. So before going to the actual topic, let us discuss few terms. First is genetics. What is genetics? It is the study of genes, genetic variation, and heredity. Means how traits are passed from parents to offspring. That is genetics. Now, when we say bacterial genetics, so we'll be dealing with the informations related to how bacteria can pass its genetic information and what is the genetic makeup of a bacteria and how these informations are passed from one bacterium to another. So all these processes will be discussed under bacterial genetics. Now, what is a gene? A gene is the functional unit of heredity. A gene is the functional unit of heredity, a segment of DNA that contains instruction for a specific character or trait. And this character or trait can be passed from parent to the offspring. That is the gene. Now, genotype. What is genotype? Genotype is the genetic constitution of an organism and they are stable and heritable. Means they can pass from one generation to another. And they are not generally influenced by the environmental factors. That is the genotype. Now, again, what is phenotype? So, phenotype is the expression of genotype in the observable properties. And phenotypic variations, they are influenced by environmental factors and they are not heritable as well as they are temporary only. Okay. So, now why this bacterial genetics is important to understand. This genetic study, bacterial genetic study, it is generally it is used as a model to understand the DNA replication, different genetic characters, and different changes, and they are transferred from one generation to another. So there are certain reasons why microbes are chosen for genetic studies. Okay. Again, it has some medical and health implications. We can uh, study antibiotic resistance and other related uh, factors, gene variations or mutations. This can be studied by using microbial genetics. And it has some implications in biotechnological and other scientific advancements also, because so many discoveries has been made or so many advances in the scientific arena has been made by using genetic engineering. Okay, genetic engineering, here bacterial genetics is the basis for genetic engineering. So for all these reasons, study of bacteria or understanding of bacterial genetics is very important. Now, the main reason of discussing this topic of bacterial genetics is to understand the mechanisms how these different types of variations are generated in a bacteria. So in bacterial genetics, mostly all those mechanisms will be discussed through which there are bacterial in a bacterial population genetics variations occurs. Okay, so generally genetic variations in case of bacteria, as we have already discussed, these genotypic or genetic variations, they are stable variations, they are heritable, and they are not influenced by environmental factors. So in bacteria, mostly it occurs by two ways. First is the mutation, then second is the bacterial recombination. So again, bacterial recombination, there are three different mechanisms are there, namely transformation, transduction, and conjugation. So broadly, there are two ways. One is mutation, second one is bacterial recombination. So bacterial recombination may occur through three different methods. One is transformation, second one is transduction, and third one is conjugation. So let us first discuss mutation. What is mutation? So mutation is the change in nucleotide sequence of a gene. Okay, it is when we say it is mutation, so means it is stable and it is heritable. Then only it will be said as mutation. So when a bacterium or organism it carries a change in its genetic makeup, that bacteria is called as mutant. It is called as mutant variety. So mutation in bacteria it may occur by two ways. First way is the spontaneous mutations. So spontaneous means it can occur automatically without any effect of external factors. So these are spontaneous mutation. Then second way is the induced mutations. So these are purposefully mutation or changes are induced in a particular gene. So this there may be exposure to the mutagens. Mutagens are the substances which can cause mutations to occur. So there may be exposure to mutagens or which may be a physical or chemical agent. Okay, these are induced mutations. Now there are different types of mutations are there in bacteria. So most common types one are point mutation and second type is the frame shift mutation. So first let us discuss the point mutation. So this point mutation it occurs as a result of substitution of one nucleotide for another. Okay, means there is only change in one single nucleotide. So there may be transition type when a purine base is substituted with another uh, purine, or a pyrimidine base is substituted with another pyrimidine. So this type of changes are called as transition type. Sometimes there may be transversion type. Transversion type means what? So here purine base will be replaced by a pyrimidine base. So that is the transversion type of point mutation. Okay. Next, again, point mutation. There are different types. First type of point mutation is the silent mutation. Silent mutation. It is also called as synonymous mutation or neutral mutation. So what happens here? There is change in the nucleotide sequence of a codon, but there will be no change in the amino acid coded by that codon. So what happens here? Only one nucleotide will be changed. There will be change in the only single nucleotide, but there will be no change in the amino acid coded by that codon. Okay, means there will be no effect in the due to the change. That is why it is called as silent. You cannot really recognize that changes has occurred. So that is why it is also called as synonymous mutation or neutral mutation. So in the figure you can see there is substitution of A with Z. So this substitution has resulted in changing the codon UGU to UGC, but still both these codons they signifies the same amino acid cysteine. So that is why there is no change in the change has occurred due to the mutation. So as there is no visible change, this type of mutations are called as silent mutation or synonymous mutation, or they are also called as neutral mutation. So next type is the missense mutation. So here what happens? Here again single base substitution will be there, but here there will be sense in the codon. So one nucleotide base will be changed or replaced by another, but in this case the codon which codes for an amino acid will be changed. Okay, and due to these changes in the codon, so a new or different amino acid will be signified by the new codon and resulting in visible changes. So this type of mutations are called as missense mutation. Okay, so in the figure you can see C is replaced here by T, and because of replacement of C with T, there is change in the codon. Initially it was UGU and later on it was changed to UAU. Okay, and these codons they signify a different amino acid. So in the first case it was cysteine, and in the second case it is tyrosine. So this is missense mutation. So next type of mutation is the nonsense mutation. So nonsense mutation, here what happens? Single substitution occur causing mutation, but due to that changes or replacement in a single nucleotide, there is generation of a stop codon. So there are three stop codons: UAA, UAZ, and UGA. These three are stop codons. So whenever these stop codons will come, so there will be immediately termination of the polypeptide synthesis. So what happens here? When there will be change in the single nucleotide due to mutation, so it leads to generation of a stop codon. Okay, so all of a sudden the stop codon will be generated and there will be premature termination of the polypeptide chain during translation. So in the diagram you can see T is replaced, A is substituted with T, resulting in a stop codon UZA. This is a stop codon. So what happens here due to nonsense mutation? So as this polypeptide chain is terminated prematurely, so it will result in formation of short or incomplete or mostly non-functional polypeptide. Okay, this is the result of nonsense mutation. So second type of mutation is frame shift mutation. First type was point mutation and second is frame shift mutation. So from the name itself you can understand. So here also there is addition or loss of one or more nucleotides, and this occurs within the coding region of a gene. Okay, and when this addition or deletion occurs, there is change in the coding sequence of that. The coding sequence of the gene will be changed, or there will be shifting of the coding region. Okay, so it may be either insertion or deletion. As I said, there may be addition of new nucleotide, that is insertion, or there may be deletion of a specific nucleotide. But whatever the form may be, due to addition or deletion, so there is change or shifting of the uh, triplet codon, means the reading frame of the polypeptide will be changed, resulting in a different generation of a different codon. Okay, and finally what will happen due to changes in the frame shift or reading frame, the shifting due to shifting of the reading frame, so it will lead to generation of a different codon, and finally a different, completely different polypeptide will be synthesized. So normally, as this polypeptide is a completely new polypeptide or different polypeptide will be synthesized, so this generally results in a generation of a non-functional polypeptide. Okay, so result of frame shift mutation generally It is a it is it leads to change in the reading frame, okay, and results in generation of non-functional polypeptide. Now, second type of generation of genetic variation in bacteria is the genetic recombination. So there are different ways or different methods by which genetic recombination can occur in bacteria. So genetic recombination, what is this? This is the formation of a new genotype by reassortment of genes following an exchange of genetic material between two different chromosomes. This is genetic recombination. Now, in prokaryotes, genetic recombination occurs because of fragments of homologous DNA from a donor chromosome are transferred to a recipient chromosome. So this is the main mechanism uh, in prokaryotes. So in bacteria, it is said as this is horizontal gene transfer. So vertical gene transfer, you all know, it is the transmission of genetic information from parent to the offspring. Okay, this is the vertical transfer. But in case of bacteria, the genetic information can be transferred horizontally. Okay, horizontal gene transfer, what is that? They can pass their genes not only to their offspring, but also they can uh, transfer the genetic information laterally, okay, to other microbes of the same generation. It is not required that it should be a parent and offspring, but to other organisms of the same generation, they can transmit laterally from one organism to another. So because of this, it is called as horizontal gene transfer. So recombination in case of bacteria, it may occur through three major methods. First method is the transformation. Transformation is the transfer of bacterial genes involving free DNA. So here, naked DNA is transferred from the donor bacterium to a recipient bacterium. Second method is the transduction. This is transfer of genetic information from one organism, one bacteria, one bacterial cell to another. But in this case, it is mediated by a virus which infects bacteria. These viruses are called as bacteriophages. Through this bacteriophage, there is transfer of the genetic material from one bacterium to another. So this method is called as transduction. Then third method is the conjugation. Conjugation is what? It is the transfer of genes from one cell to another through cell to cell contact. Okay, these three are the main uh, ways by which there may be bacterial genetic information. There is recombination in bacteria may occur. Now, first let us discuss the transformation process. So, transformation is a method of genetic recombination in which a naked DNA or cell-free DNA containing the genes on a segment is transferred to a competent recipient bacteria. Okay, so here naked DNA is transferred from one organism, one bacteria to another. Okay, so this method was discovered by Frederick Gift in the year 1928 through his beautiful experiment on the bacterium Streptococcus pneumoniae. So what he performed to demonstrate the genetic transformation? So Streptococcus pneumoniae, it has two different variants. One variant is the capsulated variant. It is also called as smooth variant, and other variant is the non-capsulated variant, otherwise also called as rough variant. So the capsulated variant or a smooth variant, they are virulent organisms, but this rough form or non-capsulated organism, they are non-virulent. Virulent are smooth forms are virulent and rough forms are non-virulent or avirulent, we can say. So what Griffith did, he injected a group of mice with smooth form. Okay, so in the diagram you can see. So in the first uh, experiment, what he did, live smooth strains which are virulent, these strains were injected to mice. So as we have discussed that these are virulent organisms, so it caused death of the mice. Then in the second step, what he did, he took live organisms of rough variant, which is known to be avirulent, so he injected to mice. So in this case, as expected, there was no death of mice was recorded. So in the third phase, what he did, he he killed the smooth variant, which was known to be virulent. He killed by applying heat. These organisms were killed by applying heat, and those were injected to mice. So in this case also, what happened, there was no death of the organism. But in the fourth, actually, it was a very critical observation. So what he did, he mixed the heat killed smooth variant, means virulent variant, with live avirulent strain. He mixed both the cultures and injected to uh, mice. So what happened, to his surprise, in this case there was death of the mice. So why it occurred? So he concluded that the normally see this heat kill virulent organisms were heat killed in the last phase. Virulent organisms were heat killed. They should not cause death of the mice. Only the avirulent strain, that is the rough strain, they were live, and they cannot cause disease or they cannot cause death of the mice. But here, as both the rough strains, virulent and avirulent strains were mixed, there is death of the mice. And Griffith could recover the virulent strains of Streptococcus pneumoniae from the dead mice. So, what was the conclusion? So, the genetic material from the dead or killed virulent strain was transferred to the avirulent strain, and they transformed to become virulent, although they were initially avirulent or non-virulent. So by getting the genetic material virulent genes from the heat killed virulent organism, so they attain the property of virulence, and they could uh, cause death of the mice. So this was a very classical, beautiful experiment explaining the transformation process. This was conducted by Griffith in the year 1928, and based on this experiment, it was proved that this genetic transformation in case of bacteria occurs, and there is transfer of genetic material or the DNA from one organism to the one bacterium to another can take place. Next method is the transduction. So here what happens, as we have already discussed, here this is also a transfer of genetic material from one bacteria to another, but in this case this is mediated by a virus which can infect bacteria. These viruses are bacteriophages. Bacteriophages are the mediator. In this case, the transduction is the process of transfer of the genetic material from one bacterium to another through a bacteriophage. Okay, so bacteriophage, what happens? Uh, they have two different life cycle, two types of life cycle. One is called lytic life cycle, also called as virulent life cycle, and second type is the temperate or non-lytic life cycle. Okay, so in the virulent life cycle, what happens? Large number of progeny phases are built up by replication inside the host bacterium, and the host cell, the these progeny viruses, they can cause lysis of the bacterial cell to get the new progeny viruses released to the environment. So this is called as virulent bacteriophages. These phases are called as virulent bacteriophages, and the process is called as lytic cycle. Here, lysis of the bacterial cell occurs. The second type of life cycle, it is the temperate or non-lytic cycle. So what happens here? So in this cycle, the host bacterium is unharmed. There will be no harm to the caused by the virus to the host bacterium. Instead, the phage DNA, it gets integrated with the bacterial chromosome, and this integrated uh, phage DNA is called as prophase. Okay, it is integrated to the bacterial DNA as prophase, and it is replicated continuously along with the bacterial cell, a bacterial DNA replication. And this through this replication process, it is transferred to the daughter cells. When new cells will be formed, so this prophase will be transferred to the next generation. So this process is called as lysogeny. This process is called as lysogeny, and those bacteria which can harbor these prophages, they are called as lysogenic bacteria. Okay, and this lysogenic life cycle, it is lysogenic conversion. That's bacteria when it can uh, carry the lysogeny or prophase, it is called as lysogenic conversion of the bacteria. So transduction is generally of two types: generalized transduction and specialized transduction. So generalized transduction, it was discovered by two scientists, Zinder and Lederberg, in the year 1951, and this type of transduction occurs during the lytic cycle of a bacteriophage, mostly the virulent bacteriophage, but sometimes this may also occur in case of temperate phages. So here what happens? This starts with the infection of a bacteria by a bacteriophage, and once the infection is initiated from the bacteriophage, or when the bacteriophage infects the bacteria, so there is fragmentation or lysis of the bacterial DNA. Okay, and the phage machinery, it utilizes the entire bacterial machinery for production of the new progeny phases. So when these new progeny phases are uh, uh, assembled, so some portions of the fragmented bacterial DNA is picked up in the progeny phases, and when these progeny phases are released uh, by lysis and infects a new bacteria, so parts of bacterial genome is transferred along with the new phases. So this type of uh, transduction is called as generalized transduction. So here in this method, the main role is played by the bacteriophage, and any fragment of DNA, bacterial DNA, can be transferred from one bacterial cell to another. Okay, so next type of transaction is the specialized transaction. So here what happens, only certain part or certain donor genes can be transferred to the recipient. It is highly specific. A specific portion of the DNA will be transferred from one bacterium to another. This is also mediated by phases, but here in this case, the other life cycle of bacteriophage is used, that is the lysogenic life cycle in case of the temperate phases. So, as I said, in this specialized transaction process, a specific portion or certain uh, specific genes will be transferred from one bacterium to another. So because of this, this is also otherwise called as restricted transaction, uh, or it is also the specialized transaction is also called as restricted transaction, because some particular specific portion of the bacterial DNA is transferred through phases to the next or new recipient bacterium. So this is the diagrammatic illustration of the transaction process. It is mediated, as we have discussed, it is mediated by bacteriophage, and both generalized transaction and specialized transaction has been illustrated in the diagram. Okay, next method of bacterial recombination is the conjugation. So conjugation, it is the transfer of DNA by direct cell to cell contact. Okay, so this depends on the presence of a specific plasmid. This is called this conjugative plasmid. So it was first described by two scientists, that is Lederberg and Tatum, in the year 1946 in a strain of Escherichia coli. The strain name was K12. So these conjugative plasmid, these are also known as fertility factors or sex factors. It is commonly written as F factor. Okay, and these factors, they carries the genetic information which is required for conjugation process. So those bacterium which contains the conjugative plasmid or F factors, they can only act as donors, and they are written as F plus, means they have the fertility factor, and those uh, do not have, or those bacteria lacking the sex factor or fertility factor, they can act as recipient bacteria. So these conjugative plasmids, they direct the formation of a sex pilus, okay, which uh, when two bacterium they come uh, close to each other, so there is formation of a sex pilus with the help of conjugative plasmid, through which the exchange of genetic material takes place from donor bacterium to the recipient bacterium. So this factor, F factor, it is an episome and it exists outside the chromosome, or sometimes it may remain as a integration or integrated. So first type of conjugation is the F plus and F minus mating. So here only the plasmid transfer takes place, and this type of mating is always unidirectional mating. So here what happens, when a bacterium having the fertility factor or F plus bacteria or donor bacterium, which uh, come close to a recipient bacterium or F negative bacterium. So what happens, there is uh, formation of the sex pilus due to the presence of the F factor. Okay, and through this sex pilus actually there is uh, establishment of contact between the two bacterial cells. Both donor and the recipient will establish connection through this sex pilus. So this sex pilus, when it forms, the one strand of plasmid from F plasmid from the donor bacterium will be transferred to the recipient bacterium. And when this one strand is transferred, the complementary strand to the transferred strand will be synthesized by a specific DNA replication mechanism that is called as rolling circle mechanism. And through this mechanism, so both the strands, the complementary strands will be completely synthesized. And once it is completed, the F negative bacterium, which was the donor or recipient bacterium, so it will finally get converted to a F positive bacterium. Okay, this is the first type of conjugation, F plus and F negative, F positive or F negative mating. So as I said, as here only the plasmid transfer takes place. Okay, so this is the diagrammatic illustration of this mating. Then second type is the HFR conjugation. So here the uh, part of chromosomal DNA also sometimes get transferred. So what happens here, when the F factor in case of F positive bacterium, it remains as an episome. Okay, so this episomal the F factor, when it remains as an episome, it can integrate to the chromosome. Okay, those uh, bacterium where F factor remains as an episome, these bacterium are called as HFR strain or high frequency recombination strains. So what happens when conjugation occurs uh, between one donor HFR cell and F negative cell? HFR organisms or the donor organisms chromosome, part of the chromosome, gets transferred to the recipient bacteria, recipient organism, because the plasmid F factor, it remains as an episome or integrated in the HFR strains. Okay, so what happens by conjugation with HFR cell, an F negative cell may acquire new versions of the chromosomal gene, because chromosomal genes are transferred here, but it will remain F negative only at the end of the process, because it has not received the actual fertility factor. Instead, it has received the chromosomal genes. And because these chromosomal genes are transferred and F uh, fertility factors are not transferred, they will remain as F negative at the end of the process. Next type of conjugation is the F prime conjugation. So when here also F factor or fertility factor, it remains as an episome. Okay. So what happens? This episomal F factor, it can leave the bacterial chromosome all of a sudden, and it can resume its status as an autonomous plasmid. Originally, it remains as an episome, but it can leave the bacterial chromosome and it can start, it can resume its status of autonomous plasmid. Okay, so in this case, what happens? The removal from the bacterial chromosome to get its autonomous plasmid status, it requires excision. Okay, so during excision, what happens? An error occurs and a portion of chromosome DNA is also excised. Okay, means where it should be excised, it is not, it doesn't happen accurately, but instead wrongly some portion of the bacterial chromosome also gets excised along with the uh, along with the plasmid, and it become a part of the bacterial F plasmid. Okay, so this erroneously excised plasmid is a bit larger, and it is genotypically it is distinct from the original fertility factor, and it is called as F prime plasmid. It is not called as F plus plasmid, but instead, because it contains portion of the bacterial chromosomal genes, it is called as F prime plasmid. Okay, so when F prime conjugation occurs between F prime plasmid with a F negative recipient, okay, so it is similar to the first type, F plus and F negative mating, and this plasmid it is transferred, and again it is similarly copied through rolling circle mechanism. But here also genes which are present in the chromosome are not transferred. Only the plasmid genes are transferred to the recipient. Okay. So when this F prime factor is transferred during this conjugation, this process is called as sexduction. This is also called as sexduction, and the recipient becomes F prime, and it remains partially diploid because the same set of genes are transferred from the donor, because it was integrated along with the plasmid. So to some portion of the chromosomal genes also getting transferred, so that is why it remains as a diploid. Okay, so these are all three different methods or ways of or types of conjugation that may occur in bacterial recombination or transfer of genetic material from one bacterium to another. So with this, we have completed all the different methods of bacterial recombination, or the methods involving the bacterial genetics or transfer of genetic material from one bacterium to another. So I hope these informations are helpful for you. And if you find these are helpful, please subscribe to my channel, so that every new discussions conducted in this channel will be available uh, to you at the earliest. And uh, please subscribe my channel and put your comments, so that in our next discussions we can further improve, and we can accumulate much more required informations in the related topic. So with this, thank you very much. Thank you all. Bye bye.